Dear students, welcome to my class. So today I will discuss about the overview of cell cycle. So first of all, you have to know what is cell cycle. Cell cycle means it is the cyclic pathway by which one cell after division becomes two cells. So it's a cyclic pathway. That means it is made up of mainly two stages. One is the resting phase, that is interphase, and another is the divisional phase. So with the help of these two phases or two stages, one cell turned into two cell via cell division. So that cell cycle was first described by Howard and Craig in 1953. So they defined as the cell cycle is the stages through which a cell passes from one cell division to the next. During this phase, the cell grows and prepared for the division. So if we summarize this thing, so we can easily tell that the whole of the cell cycle is altered with the doubling of the genome. That means DNA becomes doubled. So that means each chromosome becomes doubled. That means each chromosome after doubling becomes a chromosome made up of two chromatids. So the total content of the DNA becomes doubled within a cell, and that doubled chromosome become half during the mitosis phase or M phase in the anaphase section. I will discuss it later in my next slides. So this cell cycle is totally made up of two stages mainly. So this picture showing the detailed cyclic depiction of the cell cycle made up of mainly four stages. These are G1, S, G2 and M, and this is a unidirectional cell cycle. That means cell undergo first in G1 stage followed by S, and after completing the S it goes to G2 and finally turn into the divisional phase, that is M phase. So M phase is made up of different stages. These are broadly categorized into two types, that is first is nuclear division, that is karyokinesis, and followed by cytoplasmic division, that is called cytokinesis. So these are the different phases of the cell cycle, in which way the cell growth and division takes place. I will discuss each and every steps one by one. So first is the G1 phase or gap 1 phase or post mitotic gap phase. So this is the G1 phase. That G1 phase vary its duration and it occupies up to 35 to 50% of the total cell cycle in time scale. So in this phase, few important phenomena are actually going on. These are the synthesis of enzymes which are required for the DNA replication. So DNA replication means the genome duplication. What I have already told, the genome duplication occurs through DNA replication, and that important phenomena occurs in the next stage, that is S phase. So for this DNA replication, the prerequisites were the important enzymes. Those enzymes are proteins, those are synthesized in this G1 phase. And simultaneously, synthesis of different kinds of RNA already occurred in this stage, which are needed for the transcription and translation. That means from DNA to RNA, that is transcription, and RNA to protein, that means enzymes. And to perform those events, huge amount of ATP molecules are required. So synthesis of ATP molecules also occurred in this G1 or gap 1 phases, and synthesis of raw material for the DNA, that also occurred. What are those? Those are the pentose sugar, phosphoric acid, and the nitrogen bases. These are important raw material for the DNA. So after completing those events, the size of the cell becomes increased, and finally the cell is ready to enter their next phase, that is S phase or synthesis phase. So this is the S phase. This is the S phase. That means synthesis phase, where the DNA synthesis takes place. That means the replication of DNA takes place in this S phase. That's why the name of the phase is synthesis phase or synthetic phase. So main duty or main event is here, the whole genome becomes duplicated. In case of animal cell, in this stage centriole divides, and followed by the synthesis of histoprotein also occur, because to form a chromatin fiber like DNA, histones are equally important. So histone proteins are also synthesis in this S phase. This picture showing how a single two-armed actually chromosome becomes four-armed. That means two sister chromatid made up of two sister chromatid. So this via DNA replication. In this way, the whole genome content within a cell become doubled in this stage via DNA replication. So after those events, this cell is ready to enter the next stage, that is final stage of interphase, that is G2 phase. So this is the G2 phase. G2 phase means is the last phase of that interphase, and this G2 phase is the final stage before the division. Why it is G2? That means gap 2 phase, or you may call it the pre-mitotic gap phase. Okay, so this stage is very important because it is the final stage of the preparatory stage or interphase. Here the following events are going on. First, the synthesis of tubulin protein, which are required for the spindle formation, and the synthesis of protein required for the plasma membrane formation. Cell organelles are doubled, that means replicate in this stage, and a lot of ATP molecules required for the movement of chromosome from equator to the pole, and RNA synthesis also takes place in this G2 phase. So after completing those events which are occur in the G2 phase, the cell is now more bigger and ready to enter the next phase, that is the divisional phase or M phase or mitotic phase or mitosis phase. So after interphase, the cell entered into their divisional phase, and the divisional phase is made up of different stages. First, the nuclear division takes place, followed by the cytoplasmic division takes place. So nuclear division takes place through 4 stages. What are those? First prophase, then metaphase, then anaphase, and then telophase. I will discuss all those stages in my next lecture, which will be going on upon the cell division. So the prophase, metaphase, anaphase, and telophase, those are the 4 steps by which karyokinesis takes place, followed by cytoplasmic division takes place, that is called cytokinesis. So the type of cytokinesis is actually 2 types, because in case of animal cell, this kind of cytokinesis takes place. That means through contractile ring formation, invagination occurs, and that one cell becomes 2 cell via cytoplasmic division. But in case of plant cell, what happened? This region, that is in the equatorial region, the cell plates actually occur, and via cell plate formation, the cytokinesis actually takes place. Okay, so this kind of cytokinesis occur in case of plant cell. So this is the basic difference between the cytokinesis of plant cell and animal cell. One of the important thing is that G0 stage. So what is G0 stage? You may heard that sometimes the cell division becomes off. That means one cell cannot go another round of cell division or another round of cell cycle. Why it is those? Because you know that to continue or to run the cell cycle, lot of ATP molecules are required. That means lot of energies are required to continue the cell cycle. So sometimes what happens, the cell cannot actually the growth factor or the energy, or you may call that in the medium in case of prokaryotic cell, that is not available enough to perform another round of cell cycle. So in such a situation where the nutrition is not available enough to run the cell cycle, the cell remain arrest, the cell cycle remain arrest, and that arrested stage is called G0 stage or quiescent stage. That means where the cell cycle remain arrest due to some external growth factor or due to some nutritional deficiency, that in that position, in that point within a G1 stage, the cell cycle remain arrest. That is particular stage is called G0 stage, or it may also call that quiescent stage. Here the metabolically the cell is active, but mechanical action is tends to 0. But if the nutrition again available in this medium or in the external environment, so the cell cycle again remain enter into a G1 and resume the growth, and the another round of cell cycle may take place. So this is called the G0 stage or quiescent stage. So this is all about the different stage and the significance of G0 stage. So I hope you will understand my class. If you have any kind of doubts or any kind of query, you may contact me through my mail. I will actually giving this mail, and you, if you have any query, you can write in my comment box. Okay, thank you very much. Thank you.